From Black Death to cholera and syphilis to typhoid, microbes have been responsible for some of the world's most devastating diseases. But at the same time, these tiny single-cell organisms have aided our survival on Earth, helping us produce some of our favourite foods. Though humans didn't realise it at the time, microbes were being put to work over 10,000 years ago in ancient Egypt to create one of our most popular beverages. Today, beer production is a multi-million-pound industry. It's drunk by over 1 billion people worldwide, who consume more than 133 billion litres of beer each year. Yet if it wasn't for microbes, there'd be no beer.
Beer is a very ancient drink. In fact, we think it first emerged at about 6000 BC, and we find evidence of this in ancient Egyptian hieroglyphs. And we know that the Egyptians took their beer extremely seriously. Some of the hieroglyphs show that they gave names like Joybringer and Heavenly to their beer.
Like beer, the Egyptians also enjoyed their wine, again highly dependent on our friendly microbes.
One inscription found in a tomb made me laugh. Give me 18 cups of wine, because I want to drink until drunkenness. My insides are like straw. I guess it was not a very good idea to deny an Egyptian a drink.
Some historians have speculated that the ancients stumbled across the power of microbes to create beer when a piece of bread, which contains the yeast needed to start the fermentation process, fell into a vat of soaking grain. Left to sit and heat up, the soaking grain germinated, producing tiny sprouts, the perfect food for the yeast enzymes. And as they munched away, they produced a kind of alcoholic gruel, which made life a whole lot better.
Egyptian beer was very different from the beer that we know. It had about 10% alcohol content, so it was quite strong. And we know what it was made of because the Egyptians were very methodical at writing down how they made their beers. In the 1980s, people were able to recreate the beer of Tutankhamun based on hieroglyph information. And it was a very different beer, actually a rather unpleasant taste because it was a mixture of hops and honey and all sorts of ingredients that are quite alien to us in terms of a beer.
And their methods of drinking beer might get some strange looks in a modern English pub. In some tomb paintings, Egyptians are seen drinking their beer from ceramic cups through tubes, somewhat like drinking with a straw. The perforated straw filtered out the solid bits from the drink. So, how do you brew the perfect beer?
Different kinds of yeast have different optimum environments and produce different kinds of beer. The bottom fermenting lager yeast prefers lower temperatures and ferments more slowly. The top fermenting ale yeast gathers at the top of the fermenting vessel and prefers higher temperatures and works quite vigorously.
You can choose your preferred taste with different types of malts, grains and hops, or even the addition of fruits and spices. But after that, it's all down to the yeast microbes. It was the invention of the microscope in 1683 which allowed Dutchman Antony van Leeuwenhoek to discover microbes. Using his own homemade microscopes, the so-called father of microbiology was the first to observe and describe single-celled organisms, which he originally called animalcules. Today, less than 5% of all known microbes cause disease, but back in the 14th century, the bacterium microbe that causes bubonic plague, or the Black Death as it was also known, became the deadliest microbe of them all. Black Death is spread by the microbe Yersinia pestis, invading and growing inside rats, fleas, cats, dogs, farm animals and humans. It thrives at around 37 degrees Celsius, the exact temperature of a healthy human body.
The way in which Black Death works is rather gruesome. The microorganisms divide inside the flea. It causes a blockage in the stomach of the flea, and then the flea gets hungry, and the starving flea goes after a human being for a drink of blood. And in the process of trying to drink the blood, it regurgitates the microbes into the bloodstream of the unfortunate Black Death victim.
In the victim's bloodstream, the invading microbe is detected by the body's immune system, which launches white blood cells in defense. But instead of the bacterium being killed, it multiplies inside the cell. Eventually the white blood cell dies, and the Yersinia pestis bacteria flood out stronger than before. More white blood cells attack, but now Yersinia pestis is powerful enough to paralyze them.
It starts off with a sneeze and a fever. It's a bit like flu, and perhaps the person who got it didn't think there was much wrong with them. But then you start to get these black lesions on your skin called buboes. In fact, that's how the disease gets its name, Black Death. And these buboes appear under your armpits, in your joints. It's excruciatingly painful. These people are forced into bed, and eventually these buboes begin to exude a pus from inside them. And you die a slow, excruciating death, maybe over many days, and very, very few people recovered from the disease.
The major 14th century outbreak of the Black Death originated in Mongolia around 1330 and quickly spread along trade routes through Asia and into Europe.
In just 4 years, from 1347 to 1351, the plague killed around 75 million people worldwide, including 1/3 of the entire population of Europe.
It reached England in the summer of 1348. Church records show that between 1348 and 1350, around 40% of the English population died.
Black Death was a very egalitarian disease. Everyone from the poor to the rich was affected. And even Princess Joan, King Edward III's favourite daughter, was killed by Black Death.
It affected young and old, men and women, rich, poor, and the clergy, in rural and urban settings alike.
One of the interesting social implications of the Black Death is that church leaders were not able to provide any explanation for how this disease was caused. And as a result, the authority of church leaders began to be eroded, and people began to question whether they should even listen to religious leaders at all. And some people have even said that the English Reformation was caused by the Black Death.
With whole villages abandoned, a new class of entrepreneurs began to take over neighbouring lands left idle, spending their newly acquired wealth on distinctive buildings and memorials. Ties to land and landlord were breaking up. From Yersinia pestis and the Black Death, a new social order was emerging. Black Death was undoubtedly one of the world's most deadly microbiological invasions.
In their ignorance, people tried all sorts of ways to fend off Black Death, such as using pouches of sweet-smelling herbs. But of course, in the end, none of these things would save them.
We all fall down.
Microbes are responsible for creating some of our most enjoyable food products, such as cheese, bread, yogurt, and of course, beer. But other microbes, such as Salmonella, Listeria, or E. coli 0157, can do us a great deal of harm. In the UK alone, there are more than 10 million cases of gastroenteritis each year, a concoction of symptoms such as nausea, vomiting, diarrhea, and stomach pain caused by eating and drinking food contaminated by microbes. Since ancient times, we have done our utmost to keep them out.
If you leave fresh food lying around, one of the first things that happens is that microbes land on it, and they start to deteriorate the food over time. And one of the worst things they can do is start to produce toxins that can be poisonous to anyone eating that food. And the ancients discovered a very, very long time ago that one way to stop this is to try and preserve food.
They used a variety of methods, such as salting, drying, icing, pickling, smoking, and fermentation for beer, cheese, and bread, all of which we still use today.
You can deny microbes water by turning the food very sugary, for example, making jam. Or we can put salt on our food, and that's why people used to salt meat in the days before refrigerators to stop microbes from growing. One of the characteristics of microbes is that they're very sensitive to temperature. Many of them don't like cold temperatures, many of them don't like hot temperatures. And so we can preserve food by changing its temperature. And one way in which we do this is to put food in a refrigerator. We also boil food. That's a very common way to preserve it. And the way in which that's working is to kill microbes that can't really grow at high temperatures.
Boiling doesn't kill all the microbes, just most of them. Some bacteria are actually resistant to the temperature of boiling water, which is 100 degrees Celsius. To get rid of them, you need to raise the temperature to about 121 degrees, but you can only do this by heating water under pressure. A common method of food preservation using this technique is canning, which was developed by the French confectioner Nicolas Appert. In the 1790s, a pair began experimenting with ways to preserve foodstuffs, succeeding with soups, vegetables, juices, dairy products, jellies, jams and syrups. He placed the food in glass jars, sealed them with cork and sealing wax, and placed them in boiling water. And then there's alcohol and acid.
Many microbes can't survive alcohol, whereas yeast can, which makes it an effective form of preservation. Acid conditions, such as vinegar or pickling, are also inhospitable environments for microbes.
Pickling to keep out microbes is responsible for some of our most famous brands. In 1922, in Burton, Staffordshire, Cross and Blackwell produced the very first jar of Branston pickle. 28 million jars of it are now sold every year, still keeping out the microbes as it did 90 years ago.
One thing you have to remember is that if you refrigerate food and the microbes on it, you only slow down the growth of the microbes. You don't actually kill them.
So, when you get back from holiday, all might not quite be as you left it. Microbes are the oldest form of life on Earth. They first appeared about 3.5 billion years ago and are critically important in sustaining our life on Earth today. Microbes generate at least half of the oxygen we breathe, but in 1885, the Dutch microbiologist Martinus Beijerinck discovered another critical function they performed: fixing nitrogen.
Nitrogen-fixing bacteria are absolutely essential for life because they fix nitrogen, one of the key elements, one of the key building blocks of all of our cells and all of our biochemistry. All animals and plants on the Earth depend upon it, but also human beings. The only way that we can get nitrogen is through the food that we eat that was originally fixed by the bacteria.
Although around 80% of the Earth's atmosphere is made of nitrogen, it's too stable for most plants and animals to break down. But that's where the microbes come in. Bacteria in the soil fix the nitrogen. That means they combine it with oxygen or hydrogen into compounds that plants can make use of. Some nitrogen-fixing microbes have even developed a symbiotic relationship with certain plants, like peas and beans.
Nitrogen enters the food chain as a result of microbes taking it from the air and converting it into more accessible compounds such as ammonia, which plants can then take up.
These nitrogen-fixing bacteria often live in the roots of legumes like bean plants. And the bean plants are very clever. They send out hairs from their roots that attract these bacteria in. And the bacteria essentially take up residence in the roots of these plants.
These bacteria are incorporated into root nodules. And in these special protective houses, bacteria work anaerobically to fix nitrogen into ammonia using a special enzyme called nitrogenase, which combines hydrogen and nitrogen.
In 1909, the German Fritz Haber discovered how to fix nitrogen chemically to create ammonia, leading to the production of inorganic fertilizers on an industrial scale. 1/3 of the world's population is now sustained by crops grown using artificial fertilizers. But inorganic fertilizers can be bad for the environment.
Plants that contain nitrogen-fixing bacteria can often grow in much poorer soils than other plants, so they're extremely useful for farmers. The farmers can grow the nitrogen-fixing plants, the bacteria will fix the nitrogen, and then they can come along with a plough and churn those plants into the soil and thereby enrich the soil with nitrogen. This is one way in which nitrogen-fixing bacteria are so essential for our agriculture.
Crop rotation uses the symbiotic relationship between plants and microbes to fertilize the soil naturally. It's been practiced since ancient people began to farm. Historic rotation methods are mentioned in Roman literature and referred to by several civilizations in Asia. Today, this ancient technique has become the cornerstone of modern organic farming.
So what do these nitrogen-fixing bacteria teach us? They teach us that human beings not only require microbes, they're not only useful for us, we actually depend upon them for our survival.
Microbes, mostly tiny bacteria, make up around 90% of the cells in a typical human body and 10% of our body weight. Most of them are in our gut and on our skin. Many microbes are beneficial, for example helping us to digest our food. Only a tiny fraction cause disease and are usually kept in check by our immune system. But when they aren't, microbes also help us to fight back. Bacteria cause disease when they are able to reproduce in the body. They produce harmful substances called toxins, which damage tissues and organs. But in nature, microbes can also produce agents called antibiotics to protect themselves against competitors.
It's a tough world out there, and you might think that you just see competition in the savannas of Africa, but in fact the microbes fight each other as well. And in fact, like martial arts, they have ways of fighting other microbes with particular moves. And one move they have is to produce antibiotics. These are compounds that allow them to kill other microbes and take all the food for themselves, or the resources that they need. And so the competition between microbes results in these very sophisticated antibiotic molecules.
The discovery of antibiotics and their power to fight bacterial disease began with Alexander Fleming. He observed the mold Penicillium notatum accidentally growing on a sample of Staphylococci and saw it had killed the surrounding colonies of disease-causing bacteria.
All antibiotics work by disrupting a critical function in the bacterial cell. For example, penicillin, discovered in 1928, prevents the cell from renewing its cell wall during growth. Eventually, the cell wall weakens and bursts.
By the 1950s, the use of antibiotics had revolutionized the treatment of previously untreatable infectious diseases. In 1967, the Surgeon General of the United States of America, William Stewart, declared, "The time has come to close the book on infectious diseases. We have basically wiped out infection in the United States." But Stewart's optimism proved premature.
The bad news is that microbes can become resistant to antibiotics, and they can change their biochemistry in order to adapt to these antibiotics and prevent the antibiotics from damaging the cell.
It's standard evolutionary behavior. When bacteria reproduce, chance mutations occur. Most will be useless, but sometimes there will be one that will protect the bacterium against a particular antibiotic. While most of the bacteria succumb to the antibiotic, the one that survives goes on to reproduce and replicate the resistance, and bacteria reproduce very fast. Scientists are on an endless quest to develop new antibiotics to defeat resistant bacteria, so-called superbugs, but they must be used properly.
Widespread use or misuse of antibiotics for minor infections means that more bacteria become exposed, and so there's more chance of resistance developing and spreading through the microbe world.
GPs prescribe 133 million courses of antibiotics every year. It's estimated that 50% of these prescriptions are unnecessary, and by taking antibiotics that you don't need, you could be making yourself more at risk from the disease. If you don't complete your antibiotic course, you expose the microbe to concentrations of antibiotic which don't kill it. Some bacteria, such as MRSA, have become highly drug resistant. These superbugs aren't some complex and new set of diseases, merely variations of common bacteria that we used to find easy to control.
The good news is that scientists are developing new synthetic antibiotics that target resistant bacteria.
But who knows whether one day a mutated bacterium might become resistant to all synthetic antibiotics. A superbug. Microbes outnumber all other species and make up most living matter. They're invaluable for humans to survive and thrive, and we're continually discovering how to exploit them, including genetically modifying them to produce medicines, food, and fuel. The first organisms to be genetically modified were E. coli bacteria by Herbert Boyer and Stanley Cohen in 1973. E. coli is a common inhabitant of the human colon. It's simple to grow and can be easily manipulated and duplicated. Today, bacteria and yeast, which cause so many human diseases, are being genetically altered to help cure illness.
Genetic engineering is really about adding things to microbes. It's about rewriting their instruction manuals to produce something different, perhaps something useful to us, like drugs. We're essentially turning them into tiny factories.
One example is insulin, which should be produced naturally in the pancreas to regulate the amount of sugar in the blood. When it's not produced, we get diabetes. Previously, natural insulin was extracted from the pancreases of cow and pig carcasses in abattoirs, but it's expensive and difficult to ensure an adequate supply. In the 1980s, scientists discovered how to produce human insulin by transplanting the genetic information for the human insulin hormone from a human cell into the DNA of the E. coli bacterium. This reprograms the bacteria to produce insulin in just the same way as they make their own proteins. In fact, the genetic modification of microbes is transforming our ability to combat all sorts of diseases.
A very good example of this is lactobacillus. This is a bacterium that naturally lives in your gut. It protects you against urinary tract infections and other diseases. And this microbe is now being engineered as a possible treatment for HIV.
We have high hopes for a new malaria treatment produced by genetically engineered bacteria and yeast that will be cheap enough to give to anyone who needs it in South America and Africa.
As demand increases for alternatives to conventional fuels, scientists are hoping that genetically modifying microbes could also produce a sustainable solution to our energy needs. They want to use the bacteria and yeast to break down organic waste products like the stems, leaves and husks from crops, and excrete biofuels like ethanol and butanol as their waste product.
Ethanol has been heralded as a cleaner, greener fuel than petrol, and yeast naturally produces ethanol from sugars, an ability traditionally used to ferment beer.
This has huge implications, since currently biofuel production uses starch crops like corn and sugar, which are extremely expensive. Microbes are also being genetically engineered into our clothing. Infusing clothes with bacteria or viruses may give them super properties, such as the ability to self-clean or naturally glow in the dark. So, the tag on your t-shirt may one day read 99% cotton and 1% E. coli. Millions of years before humans appeared, microbes were thriving here on Earth, yet less than half of 1% of the estimated 2 to 3 billion microbial species have ever been identified. Today, the search for new microbes, able to survive in the world's most extreme conditions, is helping us to address one of the last remaining challenges. Is there life beyond Earth? Microbes thrive in an extraordinary range of habitats, and often where no other life forms could exist. These include freezing conditions, very similar to the permafrost of Mars. This could help us prove that life may have been possible on Mars, and on other planets too.
The search for life on Mars is essentially a detective story. It's about looking for those telltale signatures of life, maybe changes to rocks, maybe parts of microbes left over, those parts of the evidence that all come together and tell us that Mars once had life or maybe even has life.
Mars and the Earth share many physical similarities. For example, Mars has deserts, glaciers and polar caps. To help us, we're looking on the Earth for the most new and different microbes living in the most extreme environments, which are similar to the environments on Mars. This way, we can study the signatures the microbes leave behind.
Scientists have found many rocks on Mars that look like they were in contact with water, and that tells us that there were environments on Mars that may have been like environments on Earth, environments that we know contain microorganisms. And this provides us with some compelling evidence that this is a planet that might once have harbored life.
We're looking for signs of subsurface microbes living there now or in the past. We're looking for key indicators associated with life, and these are the building blocks of the cell.
Once we know where to look on Earth, we can design and build instruments which can search similar environments on Mars. In fact, scientists testing equipment in similar geological conditions to Mars recently discovered a rare and complex microbial community living in blue ice vents inside the remote and frozen 1 million-year-old Sverefjell volcano in Norway. We know that Mars once had water because we can see evidence of old river valleys. But the atmosphere on Mars is much thinner than Earth, so it doesn't protect the surface from radiation from space and makes life there impossible today. The only place where microbes might still survive, perhaps in pockets of water, is deep underground, so drilling is the only option, but it might mean going several meters below the surface.
Studying rocks that have been in contact with water or studying rocks that contain microorganisms on Earth helps us develop instrumentation to go and look for this life on Mars. So it's really about gaining an understanding of where microbes are on Earth to help us look for life on Mars.
This information is helping the development of a life marker chip as part of the ExoMars rover expedition in 2018. It's an instrument incorporated into the rover, travelling across the surface of the planet, collecting and analysing soil and rock samples, and looking for specific molecules associated with life.
People get terribly excited about the possibility of life on Mars, but we have to remember it's just testing a hypothesis. It may be that we'll find no evidence of life on Mars in the most compelling places that we think it might have grown. It would be just as remarkable to find no life on Mars as to find life on Mars, because that would tell us that despite the fact that Mars was very similar to Earth in its early history, something was missing for life.